Welcome to EPG Pathshala. I am here to present my topic cancer immunology. After this module, you will be able to learn what is a cancer, what are the hallmarks of the cancer, what is the difference between the normal and the cancer cells, and how the normal cells become the cancerous cells. You will be also able to learn what is the relationship between the cancer and the immune system, and how the immune system maintains or manage or to prevent the growth of the cancer cells. Then you will be also learn what is how the cancer evade the immune system destruction so that it can grow as a tumor. And we will also learn what are the methods to treat the cancer. We will focus mainly on the immunotherapies. So let us start with the introduction. First, what is a cancer immunology? Cancer immunology is the interdisciplinary branch of science which deals with the study of interaction between the cancer and the immune cells. Okay. Then next question is, what is a cancer? Cancer term was derived from the Greek word karninos, which were defined by the Hippocrates, that is the father of medicine, and the meaning of which is crab. This may be probably because of the similar structure to the crab-like structures. Next is the cancer can be of mainly two types. One type of a cancer is called as the benign cancer, and the second type of cancer is called as the malignant cancer. The other terms for the cancer are also called as the tumor or neoplasm, that is a new growth. So the benign cancer are those type of a cancer or the tumors which do not spread, they remains only at a one site. Okay, though they are not invasive type, whereas the malignant type of a tumors or the cancer are those type of a cancer or the tumors which invade the other tissues and spread from one side to the another sites. So the next topic is the hallmarks of the cancer. In the year 2000, Hannan and Wenberg listed some essential alteration in the cell physiology of the cancer, and they listed them as hallmarks of the cancer. Later on in 2011, they also added some more characteristic which enables the cancer for the progression. Okay. So these are some of the hallmarks of the cancer. These are, for example, the first is the cell sufficiency in the growth signals. Cancer cells do not depend on the exogenous factor for their growth. They can synthesize or express cell growth factor themselves. For example, epidermal growth factors, they can express the epidermal growth factor and the receptor for them also. Second hallmark is the insensitivity to anti-growth signals. These cancer cells are insensitive to these tumor suppressive signals. They can produce normal cells may produce some of the signals which can kill these cells, so these are insensitive to those signals. Cancer can induce the angiogenesis. They can produce blood vessel themselves, so that they can provide it with the large amount of the nutrients or the receptors. And the cancer cells can evade the apoptosis by secreting anti-apoptotic signals, or they can evade the apoptosis. Then next is that these capability like cell sufficiency, insensitivity, or inducing angiogenesis or anti-apoptotic nature of this, they can initiate the replicative immortality. Replicative immortality can also be increased because of the presence of the telomerase enzymes. Next hallmark of the cancer is the tissue invasion and metastasis. Cancer cells have an ability to invade the tissues from one side to the another and spread. This process is known as the metastasis, that is spreading of the cancer from one side to the another is known as the metastasis. The various factors or the various cells help the tissue invasion of the cancers, like macrophages, M2 type of macrophages, or the lactic acid, which they help to invade the tissues. Then next hallmark of the cancer is the reprogramming the energy metabolism. Cancer cells, even in presence of adequate amount of oxygen, they undergo aerobic glycolysis, which is also known as the Barber effect. which help to increase the amount of pyruvate, and this pyruvate can be used for the other activities of the cells. Though there is a production of less amount of ATP, but still they can use for other purposes. And also in process there is a formation of the lactic acid which lowers the pH, and thus lowering the pH also help to the metastasis or the spreading of the cancer. The main important here, one more important hallmark of the cancer is the evading the immune destruction, which we will discuss later on. The cancer cells can evade the immune system by providing immune suppressive factors, so they are protective from the immune system. And the next, here the two hallmarks like genome instability and the mutations and the tumor promoting inflammation, these are the two characteristics which enable the cells to spread. These are acquired by the cancer cells during the spreading, or we will discuss later on how these characteristics are acquired by these cells. So the next question is, what is a tumor microenvironment? Tumor microenvironment, if we see here, these are the structures or the microenvironment. There are the two microenvironments I am showing here. The A, in which I am showing how the cells or the cancer cells metastasize or the spread from one side to the another, where the second or the B portion shows what are the immune cells present in the tumor microenvironment. So the microenvironment of the tumor cells mainly consist of the various types of immune cells like dendritic cells, mast cells, macrophages, myeloid derived suppressor cells, NK cells, etc. Whereas the tumor is present at a core, where these cells are present at a periphery, and the tumor is also provided a large number of blood vessels which provide nutrients to the cancer cells. Okay. So, what are the tumor antigens and how the normal self antigen becomes non-self in tumor cells? So, the tumor antigens are those substances which are expressed on the cancer cells and by which the normal cells or the immune cells can recognize the tumor cells. Okay. And these are mainly two types of the tumor antigens. The first type of a tumor antigen are the tumor specific antigens, and these are those antigens which are expressed only on the tumor cells. They are not expressed by the other cells. So these are specific or the unique to the tumor cells only. Whereas the second type of antigen are known as the tumor associated antigens. These are those antigens of the tumor antigen which can also be expressed on the other normal cells. That means tumor specific antigen are those antigen which are unique to the tumor cells only, where the tumor associated antigen can also be present on the other cells. Okay. And some of the more important tumor antigens can be products of the oncogenes, mutated and the overexpressed genes, or the antigens of the oncogenic viruses or the oncofetal antigens, or there may be altered glycolipids and the glycoproteins antigens, or there may also be testis specific antigen. Are these mainly the testis specific antigens are present or they are protected to the testis, but in some cases if there is a tumor they can be expressed on the cells. Now next question is how the normal cells becomes the tumor or the cancerous cells, or the self antigen becomes the non-self. There are mainly three ways by which the normal self antigen can become a non-self. So the first way is the mutation, second is the overexpression, where the third is the post-translational modification. In case of mutation, if the cells, normal cells, come in contact with any type of mutagens or the carcinogens, there may be some changes in their DNA which transcribes the mutated, transcribe and translate it to the mutated genes. When these mutated proteins are expressed on the surface along with the MHC molecules, they can be recognized as a tumor antigens. And the second way of the formation of the self antigens to non-self is the overexpression. Mainly normal proteins express at a lower or a higher concentration depending on the situation. So if these are expressed at a lower concentration, but in some cases if they are expressed or the overexpressed on the cells, they can be recorded as can be called as tumor antigens. For example, tyrosinase, which is expressed on the cells at a very low concentration, sometimes because of some changes or the regulatory factors, it may be overexpressed, which is called as a tumor antigens. And the third most important way is the post-translational modification. Generally, we know that the proteins undergo post-translational modification, but if there is any change or there is any lag or there is any abnormality in these post-translational modifications, the proteins which is formed is not normal. It may be abnormal type of a protein, which when expressed can be as a tumor antigen. Here you can see in first mutation there is a green proteins which are mutated and expressed, and in case of overexpression these green proteins are highly or the overexpressed. Okay, in case of first these were in normal cells, and in case of post-translational modification you can say green strip proteins which are expressing in which are in which the post-transcriptional modification was not proper, so they are now recognized as a non-self. So the next question is how the immune system protects the host body from the cancer. Okay, and if the immune system is able to protect the cancer, then why some of the animals get cancer, or in some of the animals cancer is formed? For this, we will use the two terms. The one term is the immunosurveillance and the second is the immunoediting. So first is, what is the immunosurveillance? Basically, the immunosurveillance is the mechanism or the process by which the immune system protects the body from the cancer. So it is a process by which immune system detects, identify pre-cancerous or the transformed cells as abnormal and eliminates them before they become harmful to the body. Okay. So immunosurveillance is a host protection mechanism by which the immune system prevents the metastasis of the cancer. Okay. But it is not that the immune system protects the cancer, it sometimes helps to progression of the cancer, and this is called as the cancer immunoediting. So the cancer immunoediting is a process wherein immune system functions not only as extrinsic tumor suppressor but also to shape the tumor immunogenicity. So, and the immunoediting, so in case of immunosurveillance, immune system protects eliminate the cancerous cells before they becomes the harmful, but later with the time if the immune system is not able to completely eliminate the cells, some remaining cells may become cancerous and they suppress the immune system and they escape this mechanism. So this is called the immunoediting. So the immunoediting can be divided into the three phases. First phase is known as the elimination phase, second phase is known as the equilibrium phase, and third phase is known as the escape phase. In case of elimination phase, immune cells eliminate the cancer cells, whereas in equilibrium phase there exists a state of immune or the tumor dormancy. So in the equilibrium phase the tumor remains at the dormant state, and in escape phase there is outgrowth of the tumor cells. So let us start with this diagram. So these are the normal healthy cells. If they come in contact with some of the carcinogens, radiations or the viral infections, they are transformed, and these transformed are here, these are a pink colored, but these transformed cells produce some danger signals as well as they produce or express the tumor antigens. So that means they are immunogenic. So if there is immunogenic, they can be removed easily. So by the intrinsic tumor suppression mechanism, for example repair, senescence or the apoptosis, these transformed cells can be eliminated and again the normal cells remain there. So this is the simple intrinsic mechanism of tumor suppression. Now next is the immunoediting. So immunoediting can be divided into the elimination phase, equilibrium phase and the escape phase. So in elimination phase, which is a classical cancer immunosurveillance, here these are the transformed cells which was not eliminated by the intrinsic mechanism, but they are highly immunogenic. They are expressing their tumor antigens and they are producing the standard signals, and they are surrounded by the various immune cells like natural killer cells, CD4 positive T cells or the CD8 positive T cells, macrophages, gamma delta T cells, CD8 positive CD8 dendritic cells. So that these cells produce some factors like perforin, interferon alpha, beta, interferon gamma, or by the trail or NKG2D receptors, or they may also produce the interleukin 12. So both the innate as well as the adaptive immunity coordinate to eliminate these cells. Okay. So here in elimination phase, the immune system, both innate as well as the adaptive immunity, are able to protect the body from the cancer cells as it can eliminate. So there is the intrinsic as well as the extrinsic tumor factors, tumor suppressor factors are acting to eliminate these cells. But sometimes all the cells are not eliminated from the body. Some cells, some cancerous cells or the precancerous cells remains in the body. So these precancerous cells undergo some changes, epigenetic or the genetic changes, so that they evolve themselves so that they can be protected from the immune cells or the factors or the anti-tumor factors. So these cells, okay, so here these are the three types of mainly cells. Okay, so highly immunogenic cells, pink or the red, poorly immunogenic cells, and the orange suppressive cells. So these are the three types of the cells. Okay, here come, and these are normal. But in this phase is known as the equilibrium phase. In this equilibrium phase, there is the state in which the factors secreted by the immune system as well as the factors secreted by the tumor cells are in an equilibrium state, so that the immune system is able to maintain the dormancy of the tumor cells. Here the tumor cells are not growing, but they are still there. They are in dormant conditions. So at this stage, if the immune system fails to remove these cells grow rapidly. Okay, they bring about intrinsic as well as extrinsic changes themselves, and they evolve. Yeah. In case of escape phase, you can see here. So the escape phase here, there the poorly immunogenic number of the poorly immunogenic cells is increased. Okay, number of the suppressor cells is also increased. Okay, and where the highly immunogenic cells are reduced. Okay, and now these poorly immunogenic cells have changed. Yeah, they are now and they are not expressing. So the intrinsic mechanism which takes place in these cells, there is a loss of the antigens. So first of all, they are losing their antigens. If they are losing their antigens, so they cannot be recognized by the immune cells. Okay, so and they are producing, and the suppressor cells are producing the suppressor cells are recruiting the Treg cells or the myeloid suppressor cells, so that they can produce more immunosuppressor factors, which can more and more reduce the anti-tumor activity. So here they are and they are also expressing the ligand for the inhibitory activities or inhibitory pathways of the T cells, CD8 positive T cells, or the PGL ligand for the programmed death. So, and they are also here there is a formation of the normal macrophages into the M2 type of a macrophages, and which can increase the proliferation of the tumor cells. And this diagram shows the enlargement or the role of innate and the adaptive immune system how these can eliminate the cells. So these are the first step. Mainly in innate human system, there are the three types of cells which play a major role: NK cells, dendritic cells, and the macrophages. The NK cells can recognize the loss on those cells which have even less MHC molecules. So it can kill these cells by releasing the perforins and the granzymes, where the dendritic cells can phagocytose these cells, and the macrophages, which are the M type of a macrophages, can secrete the nitric oxide and the reactive oxygen species, by which they can kill the tumor cells. But here in this case, one difficulty to activate the innate adaptive immunity is because the T cells require the MHC molecule expression of the antigen through the MHC molecules. Along with the MHC molecules, and they also require the co-stimulatory molecules. But tumor cells reduce the co-stimulatory molecules expression as well as the MHC molecules. So here it is difficult to activate the T cells. So here there is another mechanism by which the T cells can be activated. Here they are the dendritic cells. These dendritic cells can cross present these antigens to the T cells, and so that they can activate the helper T cells, and by activating the helper T cells, the T helper cells can activate the B cells to produce the antibodies, and these can activate or transform to the CD8 positive T cells, which again can produce the perforin as well as the granzymes, which can kill the tumor cells. And this is also there is a coordination between the cell mediated and the humoral immunity, which can destroy the cancer, okay, by producing some of the cytokines or like interferon gamma or the TNF beta, or they can also bring about the apoptosis of the cancer with the help of the antibodies. So now in case of elimination phase, we have seen that in elimination phase, the innate as well as the adaptive immunity interact to eliminate the tumor cells. Okay, innate immunity have the three cells, that is the dendritic cells, natural killer cells, and the macrophages. And in adaptive immunity, T cells and the B cells play a major role to eliminate the cancer cells. Okay. In adaptive immunity can also again divide into cell mediated and the humoral immunity, which coordinately remove the cancer cells. Now next phase, the equilibrium phase. Okay, we have seen that if the immune system is strong enough to eliminate all the cancer cells, so there is no cancer. Okay. But if sometimes some of the immune system is not enough to eliminate all the cancer cells, that is a partial elimination of the cancer cells, these remaining or the surviving cancer cells or the precancerous cells undergo a stage of equilibrium, where they bring about changes in themselves, or they adapt to that environment, and these evolve over the time so that they can escape the immune system. So but still the immune system is active, and it is preventing the formation of these cells. So there is equilibrium, and the tumor cells are in dormant conditions. So here the anti-inflammatory anti-tumor factors and the anti-tumor and the anti-immunity factors are in equal amount. So if those cells, those tumor variants which were formed were not eliminated but surviving, and now these tumor variants can again modify themselves by the intrinsic as well as the extrinsic mechanism. So some of the intrinsic mechanism in the tumor cells is the loss of the tumor antigen expression, that is they express a low amount of MHC molecules. So if there is a low amount of MHC molecules, so their CD8 positive T cells cannot kill these tumor cells. And then there also may be inaccessibility to the tumor antigens to the immune cells. So the immune cells or the tumor cells can be masked so that they cannot be found out by the immune cells. And there can be also absence of low expression of the co-stimulatory molecules or the class 2 MHC molecules. Okay. So because of the absence of the class 1 MHC molecules, CD8 positive cells cannot kill the cells, whereas if there is absence of co-stimulatory molecules and the MHC MHC2 molecules, CD8 positive T cells also cannot kill the tumor cells. Okay. So and there is also more some of the more mechanism like inhibition of the immune response through the checkpoints. So these tumor cells produce the ligand for the checkpoints which are inhibitory, or when these checkpoints can block the activity of the T cell. Okay. So they provide the ligand for these checkpoints, and they also secrete the anti-apoptotic molecules which prevent their death. Okay. They can also produce some of the factor which can increase their angiogenesis. They can produce IL-6 which can promote the tumor, or they can also produce some of the suppression anti-tumor factor like TGF-beta. They can produce the TGF-beta which can kill or the suppress the immune cell activity, but themselves they reduce the receptor for this so that they cannot be killed by the TGF-beta. Okay. And the extrinsic mechanism includes the tumor growth and the invasiveness which can be increased by the formation of the M2, M1 cells to M1 to M2 cells. The M2 type of cells, macrophages, these are the tumor associated macrophages which are produced during the low amount of IL-12 or the high amount of the IL-10. These M2 macrophages, they cannot produce the reactive oxygen species or the nitric oxygen species to kill the cells. Rather they promote the tumor progression. The extrinsic mechanism also includes the cytokine blockage or the cytokine block, the tumor associated dendritic cells maturation, or it can produce IL-6 or the IL-10 which can block the DC or the dendritic cell maturation. And T cell responses can also be suppressed by the recruitment of the regulatory T cells or myeloid suppressor cells, myeloid derived suppressor cells. So that when there is a recruitment of a T regulatory cells brings about the suppression of the function of the immune cells, as well as the myeloid suppressor cells also bring about the suppression of these cells. So this is the diagram which shows the escape phase, in which the first, let us see, these are the tumor cells. Okay. Red color cells are the tumor cells, whereas these are the transformed cells, and but these transformed cells are producing these are producing or expressing the antigens over there. So these are easily can be killed by the CD8 positive T cells or the CD4 positive T cells. Okay. So but if later on they recruit the myeloid derived suppressor cells, and these myeloid derived suppressor cells or MDSCs, they produce the factors like VEGF, PG2 or the IL-1 beta, which cells produce more promotion or the proliferation of these cells. Okay. And these cells produce the factors like TGF-beta, arginase 1 or ionose, which block the activity of the CD4 positive T cells. Okay. Okay. So, and these myeloid derived suppressor cells can also produce the IL-4 or IL-13, which bring about the polarization of the M1 cells to the M2 cells. M2 cells can again produce the TGF-beta, IL-10 or PDGF, which inhibit the CD4 T cell activity. Okay. And these T cells also produce the ligand for the checkpoints so that they can activate the inhibitory action of the Treg cells, or they can produce the ligand for the apoptosis for the CD8 positive T cells apoptosis. Okay. So and there is a reduction also, there is a reduction of the antigen presentation cells and increase anti-apoptotic mediators here. So the tumor cells, okay, and they also they reduce, they increase the expression of the cell promotion, and then there is a reduction of the MHC molecules. Okay. And there are the suppressor cells. These suppressor cells, which suppressor cells, they can inhibit the activity of the natural killer cells, and they can also bring about the reduce the maturation of the dendritic cells. And these cells which have reduced MHC molecules, they can also produce the TGF-beta, IGO or IL-10 or the gangliosides or the galactin-1, which can inhibit the CD8 positive T cell activity. Okay. And these tumor cells also providing the ligand or the fast ligand which can kill the CD8 positive T cells, and then also they can be killed, and they also can kill the CD8 positive T cells. Okay. So now by this mechanism, so now this is the mechanism by which the immune cells, so by the immunosurveillance mechanism, immune cell or the immune system protects the growth or protects the human body or our body from the cancer formation. But not only the immune system is protecting the body from the cancer. The cancer cells are also evolving themselves to prevent promote their growth, so that they have a mechanism to prevent the or the protect from the immune system. Now we know that the immune system can protect the cancer formation, but if immune system fails, the cancer cells undergo escape phase. That means they can grow and form the tumor cells. So in summary, cancer immunology is the branch of science which deals with the study of the interaction between the cancer and the immune cells. And the cancer is the unregulated, uncontrolled growth of the cells. And the hallmarks of the cancer includes the cell sufficiency in the growth, insensitive to the signals, insensitive to the anti-apoptotic factors, or then inducing the angiogenesis, reprogramming the cell and energy metabolism. They can evade the immune system, or then genomic instability and the promoting the inflammation. Okay. Next is how the immune system and the cancer can be of two types: benign cancer or the malignant cancer. Benign tumors remains only at one site; they don't invade the tissues, whereas the malignant tumors they invade the healthy normal tissues, okay, and then can spread from one side to the another. And the immune system protects the host body from the cancer by producing the immunosuppressive tumor suppressive factors, which can be different types of the cells or by the cytokines. And this process is known as the immunosurveillance. So the immunosurveillance is the process by which the immune system protects the body from the cancerous cells by eliminating them. If the immune system fails to eliminate all the cells, these cells or the transformed cells undergo some changes, or they evolve themselves to modify the immune system and then evade the immune system. This process is known as the immunoediting. And immunoediting can be divided into the three phases. One is the elimination phase, second is the equilibrium phase, and the third is escape phase. So escape phase is the condition where the tumor cells suppress the immune system and then themselves spread from one side to the another side. Okay. And there are the immunotherapies which can prevent which can treat the cancers, so the immunotherapy are the type of therapies which can use or enhance the immunity of the immune system of the patient, so that they can prevent or they can be protected from the cancer. And the immunotherapy can include the cell transfer or the adaptive cell transfer, T cell transfer. Thank you.